Morning, everybody. Let's do another endocrinology lecture. It is spring 2020, week 3. This is the Tuesday lecture, so I'll usually do 2 endocrinology lectures until I'm out of endocrinology slides, and then we start doing other stuff these days. There's plenty to do in this GI-GU endo class, right? All right, so last time we got about halfway through antidiuretic hormone. Remember, there's a bunch of AKAs, ADH, vasopressin, Arginine Vasopressin, Argipressin. Those are all AKAs, so you have to be familiar with those because you never know what which one of those the question's going to use. We said typically when high levels of ADH are expressed, call it Arginine Vasopressin, usually talking about vasoconstriction with that, which we'll look at a little more today. And it is the key hormone for controlling water homeostasis, as we said, signals the kidney to conserve free water. Sodium does not follow. We'll look at that more closely today. In high concentrations, it can signal the arterioles and venules to constrict, mainly of skeletal muscle and dermis and mesenteric arteries. Who makes it? It's made in the hypothalamus, specifically by magnocellular neurons. It's found in 2 nuclei in the hypothalamus. Where are these neurons found? In the supraoptic nuclei, the sun, or the periventricular nuclei. Parvocellular neurons also secrete ADH, but that's actually used to help release ACTH. That one goes to the anterior pituitary. And those parvocellular neurons are found in periventricular nuclei. All right, so in response to ADH, what do the kidneys do? They take up free water, so they conserve free water without taking up sodium, as we've said before. But another important job of ADH is to constrict blood vessels. And so we need to look at that. In someone who's bleeding real bad, you can shut off your skin, your skeletal muscle, and your intestine via the mesenteric arteries. Those all constrict because of high levels of ADH. At that point, it's usually called Arginine Vasopressin. Quick note about dehydration. ADH is also the main defender against dehydration. Now, typically mild to moderate, not severe, but just your run-of-the-mill dehydration, it causes a loss of water without much sodium loss. So there's no sense of just turning on the R2A system. If you can release ADH, you can get a reabsorption of free water to replace the water that's lost without messing with the sodium. But anyway, with dehydration, you get increased osmolarity, because if you do lose the water, that's going to increase the osmolarity, right? I don't need to do that, do I? Let me just so I could do that. Let's do that for a second. Here's a cell, or here's the blood vessel. And normally you have a certain amount of sodium to water ratio. We'll just keep it really simple. So if you become dehydrated and lose water, then all you're left with is sodium. And there's other water around, but just to make the point, the concentration of sodium goes way up. Okay, so that's osmolarity, increased osmolarity or osmolality. Or you could say hypernatremia. Those all are basically the same thing. So ADH nicely replaces the lost water without adding sodium. And this is the strongest, by the way. Dehydration or hypernatremia or increased osmolarity is the strongest stimuli for the release of ADH. What effect does it have on aldosterone and the zona glomerula? What do those granulosa cells do? Glomerulosa cells do? It doesn't. Sodium can't bind with them, so it has to turn on the R2A system to release angiotensin II, and that's how that works. All right, here's a kind of a chart of dehydration. So you lose volume, And because you lose only water, not a whole bunch of salt when it's little, the osmolarity goes up, stimulates the release of ADH, causes the reabsorption of free water. Of course, if the volume goes down significantly, you get the whole train of this stuff going on here, right? Because then we get, if you decrease volume, that turns on renin, that converts into angiotensin 2, that stimulates the release of aldosterone, that stimulates the reabsorption of sodium and water. You guys probably pretty good with that. Vasoconstriction, so as we said, the antidiuretic effects are typically seen at lower levels of ADH concentration. But if you get high levels of ADH concentration in the blood for someone who's bleeding, internally bleeding, you will get a vasoconstrictive effect. And typically ADH is called Arginine Vasopressin. You could still call it ADH, but that's kind of the way they do that. So we need to talk about how can that be? Well, there's got to be two different receptors for that to work, and that's how ADH can affect the kidney and the blood vessels kind of differently. So antidiuretic effects is accomplished by binding V2 receptors, which are located on our friends, the principal cells. And those V2 receptors, they're in the basolateral membranes, specifically in principal cells and the distal convoluted tubules and connecting tubules, and collecting ducts is where they live. And so we know we know how that story, or we don't know. We haven't really talked about that. We will look at that story in a minute. But there's V1 receptors, a different type of receptors on the smooth muscle in the arterioles and the arteriovenous shunts, AV shunts, arteriovenous shunts they're called, are also really important in the skin. But the bottom line is, so ADH can bind to V2 receptors in low concentrations on the kidney, cause the reabsorption of free water. If you get high concentrations of ADH, they can bind to V1 receptors to shut down the skin and the skeletal muscle. Okay, so these receptors, so now you're wondering, well, how come if it's at low concentrations, why doesn't it stimulate this? Because these receptors, these V1 receptors, have very low affinity for ADH. So a little bit of around, it doesn't bind. There has to be a high concentration for it to start actually binding to this stuff. Okay, here's a little cartoon. Here is a principal cell, and we could say wherever, distal convoluted tubule, connecting tubule, collecting duct, wherever. And here we have a ADH binding to a V2 receptor, and it causes through protein kinase A, it causes the reabsorption of free water only. We'll look at that mechanism more. And here we have in vascular smooth muscle, inside of a vessel, let's say it's an AV shunt, or you can say it's an arteriole in the skeletal muscle, wherever. When ADH, you notice they've changed the wording, now they call it Arginine Vasopressin. When it binds, it causes a vasoconstriction, contraction of that muscle, which can completely shut off the artery or the capillaries that it goes to. ADH in collecting ducts. So with ADH free water sucked out of the principal cells, this occurs both in the cortical and medullary portions of the collecting duct. Let's look a little bit deeper at this. And this is just a general physiology. Remember how reabsorption occurs in the nephron. Here's where the potato pickers live, right? Those are the epithelial cells of the proximal convoluted tubule, the potato pickers. Those guys have not only potatoes. In our little story, we were using sodium, but they also pick 100% of glucose. The glomerulus is like a leaky sieve, right? Glucose, amino acids, everything is dumped into there, and it picks out all of the stuff. For our purposes, we're looking at sodium. It picks out 70% of the sodium normally here. To keep the macula densa happy, it has to pick out about 70%. And that's not the only place. When we go into the thick limb, ascending limb of Henle, we pick out another 25% of the sodium there. Yeah, so that will leave, what, about 5%? That's 90, yeah, 95, about 5% of the sodium. Macula densis will sense normally about 5% sodium still left in the filtrate. If they sense less than that, right, they freak out and they indirectly trigger the release of renin. But that's kind of how the deal works there. Right, and there's just another example. I mean, we've already seen this, but I don't think I need to go through this. We are going to talk about this thick ascending loop of Henle, which has some interesting properties. And there's the proximal portion of the distal convoluted tubule is where the macula densa cells live. And there's our connecting tubule. Some people just call it the distal part of the distal convoluted tubule. There's the cortical portion of the collecting duct. Principal cells live all in this region, right? And there's the medullary portion of the collecting duct. All right, so now we have to talk about aquaporins a little deeper as well. So we talked about V2 receptors. That's how ADH binds to principal cells. But how does the water move? And it moves through aquaporins channels. And there's 4 main ones, aquaporins 1, 2, 3, and 4. And there's 3 of these are kind of the star of the show here. Aquaporins 3 and 4, these guys are always present in the bottom, the basolateral surface of the principal cell. And remember this, the bottom of the cell, that's the part that connects to the interstitium. Interstitium, of course, is where the capillaries are, right? So basolateral does not connect to the tubules where the filtrate, where the urine is. That's the apical surface. So these don't care whether ADH is present or not. They're always there. What's their job? Their job, if you have a high concentration of water in the filtrate, it'll shoot right out of there. If you have a high concentration of water that has gotten into the principal cell, it just lets it shoot right out into the interstitium and into the bloodstream. We have not got the water out of the filtrate yet. And just so, I think you guys probably don't have to draw this, but nevertheless, so let's say there's a principal cell, and here's a capillary, right? All this stuff, that's all the interstitium. And then let's make this, how about pink? Here is a tubule, collecting tubule, or proximal part of the collecting duct, whatever. The filtrate is in here. The future urine is in here. And so right here we have aquaporins 3 and 4 all the time. They're always ready to get water into the interstitium if it accumulates inside the principal cell. And then, of course, the water, once it gets in, just jumps right inside here, and it can boost your blood pressure, right? But the question is, how do we get the water out of here? And that's where aquaporins 2 channels come from. So to get the water, I guess, have I run out of colors? Guess I have. But aquaporins 2 channel is the one that inserts right here. And that's an interesting one. That one's not there all the time, as we'll see. Right? So the ADH binding to the V2 receptors, it stimulates the synthesis and implantation of aquaporins channels. And these are in these little vesicles. They float around in the cytosol. And the interesting thing is they can be reused as well. So these aquaporins 2 channels are made and stored in the vesicles, which float around the cytosol. And once the stimulus is on, there's a mechanisms or pathway. We won't get into that. But they get the message that, hey, we need to go to work. And these little vesicles dock to the apical surface, and they open up and allow water to come out of the filtrate, assuming that the filtrate is hypoosmolaric, and assuming that there's a ton of H2O molecules to the point they would love to go down a concentration gradient. So here's this kind of story here. Here's a V2 receptor, a principal cell. There's nothing bound to it. And so aquaporins channels are just hanging out inside these vesicles. So purple things are the aquaporins too. But if you get an ADH molecule that comes and binds to the V2 receptor, I guess the V2 receptor is right here. If it binds to it right here, we go through a certain mechanism of cyclic AMP. Won't get into all that. But the bottom line is these little vesicles get stimulated and they go do their jobs. Their job is to dock with the luminal membrane here, where the filtrate is, and they implant all these aquaporins 2 channels. And because the filtrate has way more H2O molecules, way more water molecules than the inside of the principal cell, this H2O just comes flooding into the principal cell. And luckily, so the principal cell doesn't explode, we have the aquaporins 3 channels on the backside here. And it'll squirt right into the interstitium, and we could draw, a blood vessel in here if we had to. But we don't, because you can imagine that. Okay, so that's the deal with those. So aquaporins 2, everything I said, with aquaporins 2 in place, the water moves from the hypoosmolaric, water-rich fluid, sodium poor, that's hypoosmolaric, into the principal cell, cytosol, and then it moves right out the basolateral surface via aquaporins 3 and 4. Got it? So in the absence of ADH, those little vesicles go and they pick up those channels and they can reuse those. So they go pick up the channels and they're quickly removed, and now we're ready to wait. And the vesicles float around and wait for another signal. So the question is, if we go back to the nephron here, how does all this water, how does this become, the filtrate become so water rich? Because, most of the sodium was picked out, and water follows the sodium out of the proximal convoluted tubules. But how does it become concentrated with water or sodium deficient? So it's very interesting. So this is called priming the pump. So ADH also binds to the cells of the thick limb of Henle, and it stimulates those. They're just like potato pickers again. These potato pickers pick sodium again out of the thick limb of Henle, more than normal. And water tries to follow, but it can't follow. That's the key. So water can't follow. So it's not that the filtrate is going to have more water molecules in it. It's going to have less sodium molecules. But nevertheless, you get a super hypoosmolaric fluid in the filtrate downstream from the thick ascending limb of Henle. And that needs to be corrected. And it's corrected by ADH implanting those aquaporins 2 channels and allowing the balance between water molecules and sodium molecules to be restored. Right? So here's a cartoon of that. So here's a receptor on these cells, and ADH binds to these V2 receptors again. And these guys, they take up sodium, which we've done. I guess I could have done it over here. But I could do it right here as well. Right? So we can make a cell right here. Let's make a big one. Here's a cell of the thick limb of Henle. Once this binds, it takes sodium out. And then water tries to follow, but it can't because there's no aquaporins 2 channels. So water bounces off and it can't. It wants to go, but it can't. So therefore, you have, past the thick limb of Henle, you have a fluid that's hypoosmolaric. It's super rich with water molecules, and that can be corrected when we get down to the distal convoluted tubules, or the places where the principal cells are. Very good with that. So everything I said, the withdrawal of sodium increases the osmotic gradient, therefore, And it encourages water to leave the filtrate when it comes across some aquaporins 2 channels. And it does that when it starts hitting the principal cells. All right, what about ADH versus sodium? So unlike aldosterone, which causes conservation of sodium water, ADH does not result in the conservation of sodium. We said that like 10 times, so make sure you know that. And it's typically set off by hypernatremia. So why would it conserve water? Yeah. So it only causes an uptake of free water. I probably could cut that slide right out of there. So ADH and smooth muscle vasoconstriction. So there's super ADH. It is the most potent, naturally occurring vasoconstrictor of vascular smooth muscle. It loves tunica media, and its ability to constrict and cause vasodilation or vasoconstriction, it's 10 times more powerful than norepinephrine and angiotensin II's effect. So it's a very powerful vasoconstrictor. And as we said before, it targets mainly blood vessels in the dermis, and it shuts down those blood vessels, and it shuts off blood flow. We've said that like a million times. Okay, immediately boosts the blood vessel. If you don't have blood going to the skin, you save a lot of blood for your other organs. If you're low on blood, why have your skin running? Why have your skeletal muscle running? Why have your gut running? And all that blood can save to boost blood pressure when you're leaking blood like crazy. It also targets the blood vessels of skeletal muscle and mesenteric vessels, which supply the large and small intestine. So yeah, that's how it restores blood volume. Remember, it's the AV bypasses, arteriovenous bypasses, and metarterials are also activated to shut off capillaries. Here's a nice example of we might have talked about this already, but this is a nice little picture of your skin. So here's the epidermis is above. It's not vascular, but right underneath we have these capillary loops which heat up the region. You get heat loss and all that. But the important point here is here's the arteriole feeding all these capillaries in the deep dermis. There's another one over here. And the arterioles, remember, they break down into capillaries. Great. Here's the vein. There's venous plexus. There's two dermal venous plexuses that drain the blood out of here. But if we want to, with Arginine Vasopressin, we can open this thing right here, and this is a AV bypass, or arteriovenous anastomosis, or arteriovenous bypass, or AV bypass, or AV anastomosis, all that combination. If that's activated, the blood does not go down the arteriole. It's pinched off there, and it goes right through this pipe directly into here's a venule right here, and yeah, it's out. So we bypassed these and basically saved all the blood from messing around in here, and a lot of blood volume can be lost if you fill all these thousands and thousands of these capillaries. Shut them off, and you can save that blood volume to keep your pressure up, to keep you alive. What about osmoreceptors? So again, the most important stimuli for the making of ADH is blood osmolarity, or osmolarity, whichever one you want to use. So if you have increased blood osmolarity, that's the same as hypernatremia. You're dehydrated. You don't have enough water molecules. You have too many salt molecules. So that makes a hyperosmolaric solution. And I think the blood gets dehydrated. That's kind of the way I do it. Or it's sodium rich. So what does that do? Well, there's two main causes of hypernatremia. So what can cause, what can cause hypernatremia with too much sodium and not enough water molecules? Well, you can either lose too much free water, or you can gain too much sodium. Either one of those will cause hypernatremia. What could cause a loss of body water? Well, excessive sweating. If you sweat a lot, especially the closer you are to the equator, add some humidity and temperature, you sweat like crazy. right? You normally lose more water than you do salt. And that's why if you ever look at sports drinks, they're mostly water. They contain a lot of water because of that. You do lose salt too. Your skin gets a little salty, but not as much water as you do. So that can cause hypovolemia if it gets bad, dehydration. Hypoaldosteronism. So if you're not secreting aldosterone for whatever reason, from some kind of cancer or something wrong with aldosterone, the glomerulosa cells, then you can't conserve salt in water, and you polyuria, you urinate too much. And then diabetes insipidus, which we'll talk about next week, that you can't utilize ADH. Well, there's many causes of it, but maybe you have a mutation to the V2 receptors, and you can't bind ADH to the principal cells, or you don't bind it very good, and therefore you lose body water. You can't reclaim that free water like you're supposed to. All right, the key osmoreceptors actually live in the third ventricle of the brain, or near that, in a region called the circumventricular organ, or the CVO. That's broken down into a subfornical organ and a organum vasculosum. Those are the two regions. I'm not going to go that deep. I'll just call it the circumventricular organ. That's a giant osmolarity receptor, or an osmoreceptor. Here it is. Here's kind of our pituitary we've been talking about, infundibular, the stalk. Here is the sun, supraoptical nuclei area. What neurons live in there? Remember? Magnocellular neurons. Mag means great or big, and they have super long wires. Their axons go all the way into the posterior pituitary. And then we have the periventricular nuclei, is near the ventricular region. And then up in the top of it, we have the circumventricular region here. And there's exactly the subfornical organ here. But those guys are osmolarity receptors. They're like macula densa kind of. They're tasting the blood for the concentration of sodium. How much sodium is there? And the receptors are in communication with small branches of the internal carotid artery. So that's one of the main pipes going up to the brain. That is the main pipe going to the brain. Vertebral arteries and basilar arteries are there too. The internal carotid artery is huge. So what is the circumventricular organ's job? It's like the macula densa, the snake tongue. It tastes the blood. It tastes its osmolarity, or osmolarity. Tastes it for sodium content. And it's super, super sensitive. In fact, a 1% increase in osmolarity will set it off a little bit. So it does not let things get out of hand. If you get a little salt in your blood, it sets off immediately, and it really causes the release of ADH, as we'll see. So the axons, where do the wires go? Who does it trigger? Well, it has axons that go to, you guessed it, the magnocellular neurons. So that's where they go. So the axons are connected to the ADH-producing cells of the magnocellular neurons. Remember, they live in the supraoptic periventricular nuclei. It's got wires going into there and gives them a nudge and say, hey, we need to get some more free water into our bloodstream, so you better release your ADH. And so hypernatremia, or too much salt in the blood, causes the CVO neurons to fire, which sends a stimulating signal to the magnocellular neurons, and they go, oh, we better get to work. And they make ADH, and they transport it down to the posterior pituitary, and it's immediately released. All right, there's a cartoon. Internal carotid artery passing by. Here's a branch of it, and it goes connected right to the circumventricular organ, and it's got a little snake tongue tasting for sodium, and now, the sodium is getting too salty tasting. We better fix that. So it sends a signal over to the magnocellular neurons, and they go, whoa, we better fix that, and they release ADH, or Arginine Vasopressin. See how all different authors call it different things. That's why you got to know those AKAs. And that, of course, is going to go bind to principal cells, V2 receptors, right, in the principal cells of the collecting ducts or connecting tubules in that region. And yeah, it's going to let the binding to V2 is going to cause the implementation of the Aquaporins channels, 2 channels, and yeah, it's going to, some water's going to go in, and you know the rest of the deal. What about the peripheral? Because I remember I learned back in the day, well, there's osmoreceptors all over your body, especially in the hepatic portal vein. And it is true, there are osmoreceptors, but they really don't do much. And there's been some research and study on that. And so in a classic experiment, if you take a small volume of hyperosmotic fluid, however you want to say it, and you inject it into the carotid artery, if you inject it into the carotid artery, the flow of blood is going to take it into the brain. The osmoreceptors in the portal system, they're not going to have any idea what's going on. And you do that, and you immediately get a huge release of ADH. And so if you do the same thing, if you injected it down in the portal system, there's a massive delay before you get the release of ADH. So it kind of calls into question whether those osmoreceptors actually work or not in the hepatic portal system. Right, so it's kind of interesting stuff. Okay, I guess we're done. We're going to start this next time. We'll just keep it. I don't overflood you. It's probably enough for today. All right, so that's the end of endocrinology until next Monday, and we'll pick it up next Monday and next Tuesday as well. All right, so get ready for the bigger CVPP lecture, which is coming up next.